Let's start this syllabus with a relaxed mindset and a boost of encouragement. We'll start with the first chapter, the cells, which is divided into 2 sections. In Unit 1.1, we will focus on the topics outlined in these specifications. Understanding the meaning of a cell, it is the basic unit of life that forms all living organisms. Cells grow, reproduce, and perform vital functions necessary for survival. Examine a light microscope and its parts before discussing its role in the study of cells. A diagram shows the key parts of the microscope, including the eyepiece, objective lenses, stage, light source, and focus knobs. A light microscope consists of several key parts that work together to magnify small specimens. The eyepiece lens allows you to view the image, while the body tube keeps the lenses aligned. The coarse focusing knob helps with initial adjustments, and the fine adjustment knob provides precise focus. The revolving nosepiece holds different objective lenses, offering various magnifications. A condenser focuses light on the specimen, and the iris diaphragm controls light intensity. A mirror reflects light onto the specimen, and the base supports the entire microscope. Lastly, the inclination joint allows you to tilt the microscope for comfortable viewing. Understanding these components is essential for effective observation and study of microscopic samples. This tool uses 2 convex lenses to make objects look up to 1500 times larger. The eyepiece lens or the ocular lens usually magnifies things by 10 times. It has a rotating nosepiece part that holds different objective lenses, allowing for magnifications of 4 times, 10 times, 40 times, and 100 times. To light up the specimen on the slide, it uses either a mirror or a bulb to direct light through the lenses, which helps enlarge the image. We can make the image clearer by adjusting the coarse and fine focus knobs. The pictures taken with the light microscope are called photomicrographs. To use a microscope, start by placing your specimen slide on the stage and adjusting the light source for proper illumination. Begin with the lowest power objective lens for a wider view, then use the coarse focus knob for rough focus, followed by the fine focus knob for clarity. If you need a closer look, switch to a higher power lens and refocus as necessary. Finally, create a detailed drawing of your observations, labeling it with the magnification used. Remember to handle the microscope carefully, clean the lenses regularly, always start with low magnification, adjust lighting for optimal visibility, and take your time for the best results. Another type of microscope is the electron microscope. This advanced tool lets us look at tiny details in the microscopic world, greatly enhancing our understanding and providing many uses in science and technology. An electron microscope is a type of microscope that uses a beam of electrons instead of light to create images, allowing for much higher resolution and magnification than regular light microscopes. It produces very detailed images, showing structures that light microscopes can't reveal, and these images are black and white, called electron micrographs, which can be viewed on a fluorescent screen or computer. Electron microscopes are important in various scientific areas, including biology for studying cell structures and viruses, materials science for exploring atomic-level properties, and nanotechnology for creating and analyzing nanomaterials. In conclusion, a comparison table outlines the differences between light microscopes and electron microscopes. Electron microscopes are much more powerful, allowing us to see finer details, while light microscopes are great for studying living organisms. Light microscopes can magnify objects up to 1500 to 2000 times and have a resolution of 200 nanometers, using visible light and glass lenses. In contrast, electron microscopes can magnify up to 10 million times with a resolution of 0.5 nanometers, using electron beams and special lenses. Light microscopes can look at both living and dead specimens on glass slides without always needing stains, while electron microscopes require dead specimens on copper mesh and always need staining, producing black and white images instead of colored ones.